I said I wanted to get into different types of exercise and how they affect gene expression. Okay, so sarcopenia, which is age-related muscle wasting, is a multifactorial process, some of which involves fat infiltration, intramyocellular lipid, meaning when you're dealing with meat, you'll see the marbleization, the fat in the meat. That is what's known as intramyocellular lipid accumulation, meaning it's fat in the muscle when you see the marbleization in meat. Sarcopenia, along with intramuscular lipids, is associated with physical inactivity, okay, which shows you that our cattle are not getting the exercise they need. Resistance training has been indicated to minimize the age-induced musculoskeletal adaptations. Thus, we aim to investigate the effects of resistance training on mRNA levels of regulatory components related to intramyocellular lipid glucose metabolism and fiber size. So, is it making the muscles bigger, and are the muscles using sugar, blood sugar, more? Okay, so let's see here. Sarcopenia, okay, we already went through this, blah, 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 okay. Let's see here. Intracellular. Evidence indicates that intracellular muscular... wait, let me see what this is. Yeah, intracellular muscular lipid accumulation, which is what IMCL stands for some reason, blunts muscle glucose transport, meaning the muscles aren't getting the energy they need to move properly. So they're atrophying in a way. Okay, so according to age-induced changes in mitochondrial biogenesis, meaning you're not making the mitochondria you used to, okay, may affect fatty acid oxidation and result in accumulation of lipids in the skeletal muscles. Okay, various transcription factors and intracellular pathways have been implicated in the regulation of fat and glucose metabolism. For example, peroxisome, proliferator activated receptor, I don't know what letter that is in Greek, but let's see here. So, blah, blah, blah. These are some of the pathways. I don't know what they are. I want to remind people, I'm not a scientist. I'm not a doctor. I'm nothing like that. And we don't need to know that for what we're doing here, because what I want to show is that exercise changes gene expression, and I don't need a PhD to show that. Skeletal muscle atrophy is a complex process that is due in part to pro-inflammatory cytokines, which includes tumor necrosis factor apoptosis inducing, and receptor Furthermore, tumor necrosis factor is what you use to kill tumors, in case you're wondering. Further downstream activation of fork head box, blah, 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 that's the FOXO, you remember me talking about that, promotes the expression of E3, let's see. Okay, here we go. Resistance training has been consistently recommended to minimize age-related muscle adaptations. In this regard, sarcopenia, along with fatty acid infiltration, is associated with physical inactivity. Moreover, it has emerged that exercise training confers beneficial effects on glucose homeostasis, meaning it helps keep your blood sugar stable, and modulates intramuscular cellular lipid accumulation. Wow, that's a mouthful. However, the effects of resistance training on molecules related to glucose homeostasis and lipogenesis, meaning the creation of fat in skeletal muscle, with advancing age are unknown. Furthermore, how resistance training affects intracellular signaling and transcription factors that control glucose homeostasis, lipogenesis, and morphology of skeletal muscle in the aging is not understood. Based on our assumptions, we hypothesize that resistance training could protect against the age-induced intramuscular cellular lipid infiltration, lipid accumulation, glucose homeostasis, and muscle atrophy via downregulation of adipogenic factors, meaning adipogenic would mean that which creates fat, and atrophy associated molecules. These are things that create atrophy. They signal for atrophy. Okay, results. Body weight in old and trained mice. Okay, the body weight of old trained mice was higher than young groups in the beginning of the experiment. Okay, so after 12 weeks of resistance training, the rats were 57.8% and 44.4% higher in comparison to their matched non-trained, the untrained mice. Body weight percentage of gain, resistance training alleviated the, so keep this in mind, resistance training alleviated the age-induced decrease of these various molecules, okay? mRNA levels of adapogenic factors in response to aging, blah, blah, blah. So in response to exercise training, resistance training elevated mRNA levels of these various molecules here. Aging did not affect these molecules. Okay, so RNA levels of atrophy and hypertrophy, related factors, blah, blah, blah. Okay, resistance training was effective at mitigating the age-associated increase in the different things that are associated, the molecules associated with, with atrophy, muscle atrophy. Interestingly, blah, blah, blah. So resistance training alleviated the age-induced increases in these molecules here. mRNA levels of glucose metabolism. Okay. Resistance training also helped to promote glucose homeostasis through gene expression. So what the point is here is that, okay, so discussion. To our knowledge, we have demonstrated, meaning the people that did the research, for the first time that resistance training lowers basically the fat in the muscle, which was associated with downregulation of various gene expressions in skeletal muscle. In addition, the results indicate that seems to transcriptionally mediate intracellular signaling related to age-dependent changes in glucose homeostasis, stasis, and fiber size. Okay. So basically, resistance training through altering gene expression helps to increase muscle size and improve glucose homeostasis. Aerobic exercise, so effects of aerobic training on gene expression and skeletal muscle of elderly men. This might be surprising to people, but aerobic exercise also helps to reverse sarcopenia. So, purpose to analyze the effect of 3 months of training on global gene expression and skeletal muscle of elderly men in order to better characterize the pathways that differentiate the trained and the sedentary state. So methods, needle biopsies were blah, blah, blah. So basically they took muscle and they looked at it. So results, overall training modified the expression of 397. See, this is the thing. People will go, oh, well, you don't know these genes and those genes. I'm not a geneticist or a biologist. There's a shitload of genes that you need to keep track of to do these things. So overall, this is why people are using artificial intelligence and stuff now, and why we need to in order to do a lot of this high-end research. So overall training modified the expression of 397 out of the 14,500 genes tested. In the trained state, the expression of genes linked with energy metabolism, protein amino acid dephosphorylation, and heme biosynthesis increased. Okay, whereas those linked with ribosome and protein catabolism decreased. So it caused an anabolic reaction. Conclusion. This set of 397 genes that includes the upregulated energy pathways and the downregulated protein catabolism genes represents the transcriptional response of the skeletal muscle in the trained state. It is highly likely that many of these genes are mediators of the beneficial effects of physical activity on health and fitness. So yes, aerobic exercise also helps to reduce sarcopenia. We're going to bring ourselves back to nutrition here, fruit and vegetable consumption and pro-inflammatory gene expression, okay? So the highest tertile of energy-adjusted fruit and vegetable consumption was associated with lower plasma concentrations of C-reactive protein and homocysteine, So essentially, conclusion, a higher fruit and vegetable consumption was independently associated not only with reduced CRP and homocysteine concentrations, but also with lower mRNA expression of a pro-inflammatory molecule there, of some relevant pro-inflammatory markers in healthy young adults. So keep that in mind. Higher fruit and vegetable consumption, lower inflammation. It's doing it through controlling the expression of certain genes. Berry intake changes liver gene expression and DNA methylation patterns associated with high-fat diet. So the liver is a critical organ for the regulation of energy, homeostasis, and fatty liver disease, is closely associated with obesity and insulin resistance. We have previously found that lingonberries, blackcurrants, bilberries prevent, whereas acai berries exacerbate the development of hepatic steatosis and obesity in the high-fat mouse model. In the follow-up study, they investigated the mechanisms behind these effects. Genome-wide hepatic gene expression profiling indicates that the protective effects of lingonberries and bilberries are accounted for by several-fold downregulation of genes involved in acute phase and inflammatory pathways. In contrast, acai berry-fed mice exhibit marked upregulation of genes associated with steatosis and lipid and cholesterol biosynthesis, which is in line with the exacerbation of high-fat induced hepatic steatosis in these mice. So basically, these mice are being fed a high-fat diet. The acai berries essentially help that along. The acai berries cause a fatty liver, whereas the lingonberries and blackcurrants decrease it. Okay. So to gain further insight into the mechanisms involved in gene expression changes, the HELP tagging assay was used to identify differentially methylated CPD. Okay, compared to we conclude that the beneficial metabolic effects of lingonberries and bilberries are associated with downregulation of inflammatory pathways, whereas blackcurrants exerting similar metabolic effects, different mechanisms of action appear to dominate. So why am I doing this, you might ask? So this is the role of phytonutrients and nutrigenetics and nutrigenomics perspective in curing breast cancer. So why am I doing this? Well, the thing is, a lot of people like to talk about, this group and that group and genetic this and blah, blah, blah. You have to understand, most people that are alive today are alive because they are evolutionary successes. Okay? A lot of the problems that we seem to be having are not due to necessarily genetic issues per se. It has to do with massive changes in our environment, massive changes in our activity level, massive changes in our diets, massive changes in the nutrient profiles of the foods that we're eating, which are causing epigenetic shifts in how our genes are expressed. Again, this is why I like talking about Dr. Terry Walls and Dr. Terry Walls' work with her work on a paleo-type diet to reverse multiple sclerosis. So breast cancer is one of the most common types of cancer and an important contributor to female mortality. Several genes and epigenetic modifications are involved in the development and progression of breast cancer. Research into phytochemistry, nutrigenomics, and nutrigenetics has provided strong evidence that certain phytonutrients are able to modulate gene expression at transcriptional and post-transcriptional levels. Such phytonutrients may also be beneficial to prevent and treat breast cancer. In this review, we will focus on the nutrigenomic effects of various phytochemicals, including polyphenols, phytoesterols, terpenoids, alkaloids, and other compounds from different sources. Overall, these phytonutrients are found to inhibit breast cancer cell proliferation, differentiation, invasion, metastases, angiogenesis, and induce apoptotic, meaning to kill cell death by targeting various molecular pathways. They also alter epigenetic mechanisms and enhance the chemosensitivity and radiosensitivity of cancer cells. Such phytochemicals may be used for the effective management of breast cancer patients in the clinical setting in the future. The present article aims to summarize the specific molecular pathways involved in the genetic effects of phytochemicals in breast cancer. Nutrigenomic effects of phytochemicals in breast cancer. Phytochemicals can be classified into different groups based on their chemical compositions, such as polyphenols, phytoesterols, terpenoids, alkaloids, and other compounds. Each of these classes have potent biological activities and can modulate various molecular targets. The present paper will blah, blah, blah. Polyphenols. Polyphenols are a broad group of organic compounds which include various bioactive plant metabolites such as phenolic acid, flavonoids, stilbenes, lignans. These compounds possess one or more benzene rings and hydroxyl groups, a wide variety, blah, blah, blah. So their health benefits include anti-carcinogenic, anti-inflammatory, antioxidant, anti-proliferative, and anti-angiogenic properties. Polyphenols and certain flavonoids are found to inhibit DNA methyltransferases and histone deacetylases, thereby enhancing acetylation and demethylation of tumor suppressor genes that prevent breast cancer proliferation and migration. Polyphenols also inhibit signal transducer and activator of transcription 3, thereby decreasing the transcription of target genes involved in immunosuppression, cell proliferation, cell survival, angiogenesis, and metastases. Okay, so phytosterol. Phytosterols are steroidal alcohols containing 28 or 29 carbon atoms, which are key components of plant plasma membrane. These are not synthesized in the human body and are mostly derived from dietary sources. Now, what's interesting is with the advent of CRISPR and things like this, never say never. Pretty soon people may be able to manufacture these things through the use of genetic modification. But that's beyond the scope of what I'm covering here. This has to do with diet. So they're not synthesized in the human body yet. A wide variety of phytosterols are present in the diet, but the most abundant ones are beta-sitosterol, campsterols, ergosterols, and stigmasterols. AMP-activated protein kinase, people will remember I talked about AMPK, is a unique target for treating many forms of cancer, including breast cancer. It acts as a sensor of cellular energy stress, thereby promoting cellular catabolic pathways. So remember, this is a caloric restriction mimetic. Remember that. Caloric restriction mimetic. Remember when I talked about the Okinawan diet and the caloric restriction mimetics. Okay. So, catabolic pathways and inhibited cellular anabolic pathways, growth and proliferation. The effects are exerted at both transcriptional and post-transcriptional levels. And people might be going, well, you're talking about exercise to promote anabolism. You're talking about protein intake to promote anabolism. And then you're talking about AMPK activators to promote catabolism. The body has a cascade of different metabolic pathways that have to be activated in order for the body to run functionally. In his book, Patrick Quillen talks about this. A lot of people are so worried about mTOR. You're going to activate mTOR. Yes, but mTOR being activated with the absence of the activation of AMPK might be a problem. However, when these pathways are made to regulate each other. So certain phytosterols can act as AMPK activators, thereby helping in tumor suppression. And by the way, read Patrick Quellen's book, Beating Cancer with Nutrition. I highly recommend that. So phytosterols also inhibit the translocation to the nucleus, blah, blah, pro-inflammatory genes, terpenoids. So basically, they knock down the pro-inflammatory genes. Terpenoids. Terpenoids are a wide group of bioactive organic compounds derived from plant sources, Based on their number of cyclic structures, they can be broadly classified as hematerpenoids, monoterpenoids, sequestroterpenoids, diterpenoids, triterpenoids, tetraterpenoids, and polyterpenoids. Their unique structures are responsible for a wide range of biological activities, such as antioxidant, anti-inflammatory, and anti-carcinogenic effects. The terpenoids are able to prevent cancer cell proliferation and induce apoptosis in cancerous cells via various complex molecular mechanisms. Higher levels of proteasomal activities can be observed in tumor cells, which degrade various proteins involved in the cell cycle. So that's basically tumor cells digest proteins. So terpenoids exert an anti-proteasomal activities, thereby preventing abnormal proteasomal functions. Terpenoids are also found to inhibit NF-κB pathway, which in turn reduces the expression of genes involved in pro-inflammatory compound production. So again, knocking down inflammation. Alkaloids are biologically active organic compounds which may act as potential anti-cancer agents. They are found to increase cytotoxicity, induce DNA damage, modulate survival pathways, increase caspase activity, promote apoptotic death. Okay, so I mean they're killing the cells, the cancer cells, cause cell cycle arrest, and may suppress NF-κB pathway, again knocking down inflammation. Nutrigenomic effects of some selected phytochemicals in breast cancer. EGCG is a unique flavonoid derived mainly from green tea. It is widely studied for its cancer-preventing properties and is known to possess antioxidant, anti-inflammatory, anti-proliferation, and anti-angiogenic, anti-metastatic, anti-genotoxic, apoptotic, why can't I say some words tonight, and epigenetic effects. It targets various key molecular pathways and modulates the expression of various genes involved in tumorigenesis, meaning the creation of tumors, and also knocks back tumor progression. EGCG is found to downregulate telomerase, help human telomerase reverse transcriptase. Additionally, it reduces the expression of beta-catenin. Genistein is a flavonoid with phytoestrogen properties. This comes from soy. Okay, so this is very important. As found in soybeans, numerous molecular pathways are targeted by genistein, which leads to its anti-proliferative, anti-inflammatory, anti-metastatic, apoptotic, and cytotoxic effects. It causes cell cycle arrest, reduces cell viability, and improves radiosensitivity. Genistein regulates ERA expression in breast cancer cells. It may repress ERA, thereby reducing cell proliferation and differentiation. It may upregulate BAX and okay, so let's go here. Quercetin, one of my favorite phytonutrients. I love this stuff. It works for me. This stuff is a miracle compared to resveratrol. Quercetin is a flavonoid which is derived from many plant sources and possesses potent anti-cancer properties. Quercetin demonstrates anti-proliferative, anti-metastatic, anti-angiogenic, apoptotic, meaning it causes cell death again, and chemosensitizing effects. It is found to repress leptin gene expression, leading to inhibition of T4/7D cell growth. The compound enhances the expression quercetin reduces the expression of FASN and B-catenin. It represses so it inhibits twist via let's get down to what it does. The ability of quercetin to target such a variety of molecular pathways makes it an ideal lead for anti-cancer drug development. I like how they always say anti-cancer drug development instead of saying take quercetin. Apigenin. Apigenin is a flavonoid present in a wide variety of fruits and vegetables, including grapefruit, chamomile, parsley, celery. Now you'll notice a lot of these are geroprotectors. Okay? Keep that in mind. A lot of these anti-cancer phytonutrients are geroprotectors, meaning they reverse the aging process. It might be important to explain that the risk of cancer goes up with the risk as you age. It may influence various molecular pathways involved in tumor genesis. Apigenin is found to sensitize. Okay. So, apigenin may increase the cytotoxic effects of a certain drug there, I guess, by enhancing DNA damage and decreasing expression of DNA repair genes. Luteolin is a potent bioactive flavone with anti-cancer properties. It is found in a wide variety of plants such as celery, parsley, thyme, chamomile tea. Okay, it reduces the expression of these molecules. Okay. These effects lead to the suppression of cell proliferation, angiogenesis, metastases, migration, and cell survival, and induce apoptosis. Camphorol is a flavonoid present in many fruits and vegetables. It has potent antioxidant and exhibits anti-cancer effects due to its anti-proliferative, anti-metastatic, apotic, and cytotoxic effects. It may downregulate insulin response. Such unique properties of camphorol may be utilized for novel drug development. Iso whatever is an active something or other. Let's see what this does here. Suppressor including okay, leading to increased thereby halting cancerous development. Curcumin, which is in turmeric, obviously, is responsible for the characteristic yellow color of turmeric. Several studies have confirmed the anti-carcinogenic, anti-proliferator, antioxidant, anti-inflammatory, anti-metastatic, anti-angiogenic, apoptotic, radioprotective, and chemosensitizing properties of curcumin. Okay. Curcumin downregulates these genes here. Additionally, research from previous years has proven curcumin may have many pharmacological activities, anti-cancer, anti-inflammatory, antioxidant, without any side effects at dietary intake levels. Whatever the heck that is a lignin, which with phytoestrogenic properties. It is biophenolytic compound abundantly present in flax seeds. Okay, this helps to increase the cytotoxic effects of chemotherapeutic agents, leading to suppression of cell proliferation, metastases, migration, cell growth, and tumor progression, and promote apoptosis and cell death. Resveratrol is a stilbene and phytoestrogen found in grapes, berries, and peanuts, and is known to possess anti-malignant properties. Again, a powerful geroprotector. So it downregulates insulin-like growth factor. Pterostilbene. Pterostilbene is a stilbene found in blueberries. The compound is chemically related to resveratrol. It may downregulate mTOR. Okay, zinc. So it may also, now that's also a very powerful geroprotector and more bioavailable than resveratrol. And as you'll notice, these are affecting you on a genetic expression level. They're changing the way your genes are expressed. Diet changes the way your genes behave. Silibinin. Silibinin is a unique flavonolignin present as an active component. Let's see, so what's this do here? It suppresses various things, okay, leading to tumor suppression. Terpenoids. Okay, this is thymoquinone, is a terpene derived from black seed oil with anti-cancer, antioxidant, anti-inflammatory, and cytotoxic properties. It downregulates these genes. Okay. Overall, okay, it's to inhibit proliferation, metastases, migration, invasion, viability, Okay, and it promotes apoptosis, necrosis, and cell death. Okay, so lactone, saponins, ginsenosides are bioactive saponins present in ginseng roots. These compounds may induce the expression of various target genes involved in breast cancer. Ginsenoside may cause epigenetic modifications of the genes involved in immune response and tumorigenesis. They may increase immune response and inhibit growth of MCF-7. A little tired, didn't sleep too well last night. Hypermethylation of certain genes such as insulin-like 5, olfactory receptor family, ginsenosides inhibit So what it does is it reduces proliferation, angiogenesis, metastases, invasion, and enhanced chemosensitivity and apoptosis. Sulforaphane, okay, it's present in cruciferous vegetables. I talked about this a lot. And this inhibits cancer stem cell increases, and that's what you got to keep in mind. These are affecting the way your genes function, causing benefits to people with cancer. So this is benefiting people with cancer by changing the way their genes are expressed through epigenetic changes. Dietary intervention by phytonutrients and their role in modulating coding and non-coding genes in cancer. So I skipped to the conclusions, but please feel free to follow this on your own. I didn't want to make my video too long. Phytochemicals are now studied as important regulators of key pathological processes, especially cancer, increasing the awareness regarding the significant contribution of phytochemicals. Furthermore, their action also consists of increased chemotherapeutic sensitivity to numerous treatment agents, improving the overall survival rate in cancer patients, passing the classical concept as antioxidants associated with natural compounds activity. Phytochemicals have been shown to regulate coding, but also non-coding genes with important significance in cancer therapy. In this way, they act as inhibitory compounds towards oncogenic coding and non-coding transcripts, and also stimulatory agents that target tumor suppressor transcripts, including miRNAs. Another important aspect consists of the capacity of natural compounds to partially reverse the MDR phenotype of cancer cells, this issue being as one of the major drawbacks in terms of cancer survival rates. Following these studies, natural agents are now exploited for the development of alternative therapeutic strategies, increasing the action of the conventional treatment schemes in cancer prevention and treatment through their multi-targeting capacity. However, the main obstacle still consists of the reduced bioavailability and stability. In order to overcome this issue, numerous attempts are made in the form of nanostructures, chemical adjustment, and synthetic production, and so on. The success of these research activities will significantly contribute to better manipulation of cancer pathologies, improving the effects of conventional therapies, but also enriching the success of prevention methods. Dietary affects gene expression and prevents lipid accumulation in the liver. This is one of the reasons I gave up a low-protein, starch-centered diet like McDougall suggested, because, to be quite honest, eating a freaking a super high carbohydrate, low-protein diet was affecting my triglycerides. Mice fed the high-protein diet displayed a lower body weight, developed less body fat, and decreased liver fat accumulation, which could be attributed to the combination of several processes, next to an increased hepatic VLDL, which is very low density and low density cholesterol production rate, increased energy utilization due to enhanced protein catabolic processes such as transamination, TCA cycle, and oxidative phosphorylation was found upon high protein ingestion. Conclusion: feeding a high protein diet prevented the development of non-alcoholic fatty liver disease by enhancing lipid secretion into VLDL particles and less efficient use of ingested calories. Okay, this is the introduction. Long-term dietary macronutrients and hepatic gene expression in aging mice. This is a meta-analysis in the review of 172 publications on the effects of nutrients on hepatic transcriptome. OSADA reported that protein intake influenced genes involved in lipogenesis, fatty acid uptake, oxidative stress, and DNA methylation. Carbohydrate influenced genes involved with oxidative stress, cell proliferation, ammonium, blah, blah, beta oxidation. Okay, by contrast, our long-term study over 15 months examining the effects of 25 different diets varying in macronutrients showed that protein intake was the most important driver of gene expression, whereas the intake of carbohydrates and fat had limited effects that were in the opposite direction. So taking in high amounts of protein is the most important factor. Fat and carbohydrate intake are the main contributors to total energy intake. Therefore, the responses to total energy intake reflected those seen for carbohydrate and fat. It appears that with aging and or very long-term dietary exposure, the only protein intake remains a potent regulator of hepatic gene expression, which presumably reflects a greater biological imperative to regulate long-term protein metabolism by the liver. It is possible that muscle and adipose tissue may play a greater role in responding to long-term changes in the dietary carbohydrate and fat, but we did not measure gene expression. You have to understand, high amounts of muscle mass help you handle carbohydrate better by burning off glucose. Because muscle is metabolically active. There have been a few other studies that have examined the effects of differing amounts of macronutrients on hepatic gene expression over longer term periods, and these reported similar effects to those we observed with dietary protein. So what about nutrients required for the proper development of the human brain and the fetus? Whereas all nutrients are needed for the development and function of the brain, certain nutrients have high impact on early brain development, including protein, fats, iron, zinc, iodine, and vitamin B12. The impact is greater in the fetal and early postnatal period because of the high metabolic demands of the brain at that age. The positive or negative neurobehavioral effects of these nutrients depends on timing, dose, and duration of provision or deprivation. Timing appears to play an important role because of the non-homogeneous nature of regional brain development and because of the unequal distribution and prevalence of nutrient deficits in a population, ensuring adequate fetal loading of nutrients through better maternal care during pregnancy and pre-pregnancy and post-pregnancy for that matter, via provision of adequate nutrition and reduction of obesity, hypertension, and glucose intolerance appears to be key to fostering postnatal nutrient sufficiency. For the term neonate, human milk provides the optimal support for neural development. Okay, whereas for at-risk populations such as preterm infants, early identification and correlation of nutrient deficits is essential to maintain the brain on trajectory through critical periods of development. So people seem to be getting stupider, right? I mean, that's obvious. If you're my age, it's obvious when you look at people. They seem to just be getting dumber. Now, you have to remember, my generation was one of the first generations that subsisted almost entirely on fast food and prepackaged foods. So keep in mind, these nutrients are especially important to the developing brain, okay? So you had so basically fats, iron, zinc, iodine, and vitamin B12. Also folate, but I'll get into that in a later date perhaps. But this is what I'm showing here is these things are required for the genetic potential to be reached in the fetus. Now I'm going to call it quits for this video, and I'm going to continue with another video in a little bit, maybe tomorrow.